Okay, now that we've got all of that terminology settled, let's talk a little bit about the evolutionary forces that change allele frequencies. And maybe the most sensible one to start with is mutation, because a mutation is the force that can generate a new allele. In other words, it can take an allele frequency from 0 to some slightly higher number. A mutation is a change in a genetic sequence. So for example, here's the base adenine changing to the base cytosine, and that can potentially change what this DNA sequence does. One thing that's very important is to distinguish mutation from recombination. Mutation generates new alleles. Recombination just shuffles existing alleles into new combinations. So for example, if you had this hand of cards, what recombination would do is shuffle this hand with another hand of cards so that you might end up with a slightly different hand. Okay, but this card, for example, the Jack of Clubs, already existed. It's just that it wasn't on this chromosome before recombination happened. So recombination doesn't generate anything new that isn't already in the deck. Mutation, though, does. Mutation is like taking the 5 of spades and it undergoes a mutation and becomes the 5 of ducks. It's something that's never been seen before. There are a number of different kinds of DNA sequences in the genome that can experience mutations. And this is important because the kind of DNA sequence that you've got, or what that DNA sequence does, really matters when it comes to what a mutation does to an organism. This is a very messy diagram, but let's just take a look at it and break it down a little bit. What you see here are certain regulatory regions. These are regions to which proteins bind during the transcription process or certain other processes, and they affect whether or not this gene gets transcribed. Okay, this whole thing gets made into RNA, and then certain parts of it get cut out. So what actually gets translated into protein are these parts called exons. Okay, so we've got 3 different types of genetic sequence here. There are protein coding regions. Those are the exons here. There are regulatory sequences, which are all these other colored bands. They have a function, but their function is not to be translated into protein. And finally, we have the gray areas in this diagram. Those are sequences that do not have a function. The most common kind of mutation is what's called a point mutation. That's when a single base pair in the DNA sequence changes. So for example, if this thymine base gets changed to an adenine base, not only is this sequence here going to be changed, but when DNA goes through replication, the strand that is copied from this original strand will also be different. And that change gets continued on down every time the cell divides. If that cell is in a sperm or an egg, it's going to pass that mutation on to an offspring, potentially. Depending on the kind of sequence that you're dealing with, mutations can have a variety of effects. Non-functional sequences don't affect the phenotype of an organism. So when you have a mutation in those regions, it doesn't change how an organism looks or behaves. The mutation will not change the phenotype. Proteins interact directly with the DNA sequence that you find in regulatory regions. So potentially any mutation to a regulatory region can change transcription. It can change when a gene is expressed, it can change how much of it is expressed, it can change the location in the body where it's expressed. Okay, not all mutations in a regulatory region do change the way this gene is expressed, but all of them have the potential to. It's a little more complicated when it comes to mutations, point mutations, in a protein coding region. Remember that the DNA code is what's called a degenerate code. What that means is that there are different codons that can code for the same amino acid. When the ribosome reads a messenger RNA and translates it into protein, remember what it's doing is it's reading 3 base pairs at a time. Uracil is present instead of thymine in RNA, so that's why you see a U instead of a T in this table. And so, for example, what a ribosome does is when it receives the RNA sequence uracil, 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 it attaches a phenylalanine amino acid residue to the protein that it's building. Okay. If this base pair in the codon gets mutated and it changes to a cytosine rather than a uracil, what the ribosome is going to read is UUC, and it's going to attach a phenylalanine residue. Okay, that's what degenerate means. It means that sometimes you can change the DNA sequence and it does not change the protein. It does not change the amino acid sequence. Okay, however, if you were to take this UUU and this last uracil were to mutate into an adenine, you would get UUA, and the ribosome would read that and put a leucine amino acid residue into the forming protein. So we've got 2 different kinds of point mutation that can occur in protein coding regions. There are synonymous mutations. That's like going from this U to this C. Both of these codons code for the same amino acid. This is called a synonymous mutation. It does not change the sequence of a protein. A non-synonymous mutation, like going from uracil here to adenine here, that does change a protein sequence because instead of having a phenylalanine in this protein, what you end up with is a leucine. Non-synonymous mutations in a protein coding region can affect an organism's phenotype. So, for example, in normal hemoglobin, in sort of the wild type allele for hemoglobin, this is the amino acid sequence. And particularly at this point in the sequence, you get a glutamic acid residue. If you change one of these base pairs, instead of getting glutamic acid, you get a valine. And that changes the whole behavior of the resulting hemoglobin protein. This is the mutation that is responsible for what's called sickle cell anemia, because hemoglobin made with this protein structure causes cells to form this sickled shape. Keep in mind that not all non-synonymous mutations that are in a coding region alter the phenotype. What's shown here is a 3-dimensional model of an enzyme. And the colors represent how evolutionarily conserved the amino acid sequences. There are some areas where the sequence is really conserved. That's the purple here. And what that means is that that amino acid sequence is the same across this wide range of organisms. The implication is that changes to these amino acids really alter the phenotype, probably by making this enzyme stop working. Because this enzyme is important for an organism's ability to survive, basically that radically changes its phenotype. It goes from having an alive phenotype basically to a dead phenotype. However, you'll notice that there's some areas that are blue. And these are places in this protein that seem to be able to change without altering the phenotype, because these parts of the protein aren't particularly important. So, not all non-synonymous mutations affect an organism. Basically, none of the synonymous mutations in protein coding regions change an organism because they don't alter the phenotype. Typical mutation rates do vary quite a bit from one part of the genome to another, but on average, there's something like 1 in a billion per base per cell division. In prokaryotes, that's bacteria and archaeans, which we'll be learning about later in the semester, the mutation rate is a little bit lower. It's about 1 in 10 billion. This varies from one tissue to another. So for human germline cells, you get about 0.06 times 1 in a billion mutations per base pair per cell division. In other tissues, it might be lower. In some tissues, it might be higher. Because there are a lot of cell divisions in a human organism between its first cell division as a budding zygote to the cell divisions that form gametes, you actually end up with about 35 new mutations in every gamete, every sperm or egg cell that gets formed. Since you inherit one gamete from mom and one from dad, that means that every new zygote that forms, or every new individual that forms, has 70 brand new mutations. This number may be of concern to you for two different reasons. You may think that it is too high, or you may be thinking that it is too low. You might be thinking that 70 is a lot of mutations, because if every individual has 70 mutations, that's changing 70 different things about each individual every time that a new baby is born. And isn't that bad for us? Well, actually, 95% of the new mutations have no effect whatsoever on the organism that inherits them. You might be thinking that this number is way too low. If there are only 70 new mutations in each individual, and 95% of them don't do anything, is that really enough raw material for evolution to work with? Because, as we'll learn later, in order to have a species keep evolving, it needs to have a lot of genetic variation. New genetic variation needs to keep arising. Well, is 70 mutations per individual enough? Even if only 1% of the remaining 5% of mutations are useful, that means that in a population of 1 million organisms, you're getting 35,000 new mutations each generation that are actually useful, that actually help improve an organism's ability to survive and reproduce. So this is actually a tremendous amount of raw material, genetic variation, that natural selection can work with. What do mutations do to the fitness of an individual? What do they do to the ability of an organism to survive and reproduce? In other words, are they helpful or are they harmful? This is data from a study that was done on some kind of microorganism. I can't remember whether it was a bacterium or a yeast or what exactly. But this is what new mutations tended to do. Most of them were neutral. So these are the mutations that were exactly neutral. That's the most frequent kind. A lot of them are nearly neutral, so they had a very, very slight harmful effect. Most of the rest are harmful in one degree or another. They have increasingly bad effects on an organism's survival and reproduction as you move this way. And quite a lot of them actually turn out to be lethal. They interrupt something that's very important for an organism. And so those mutations, if they're expressed, kill the organism. A very small percentage of new mutations are over here. Those are the beneficial mutations, the one that actually increase an organism's ability to survive and reproduce. It might strike you that this picture looks really bad. Most mutations are harmful, and many of them are very harmful. If this is the case, and if mutations are constantly happening, why is it that organisms don't just come grinding to a halt? Why is it these harmful mutations don't cause evolution to just stop? They could very well overwhelm the process. Every individual will become riddled with these difficult genetic problems to the point where a species goes extinct. The reason it doesn't is that kind of by definition, alleles that are harmful don't get passed on to the next generation. So after one generation, all of the lethal alleles, for example, are going to disappear as though they had never existed. The harmful alleles might not disappear completely, but they're certainly going to be reduced in frequency. The neutral alleles are just going to hang around. They're not going to change in frequency. As the generations go past, you're going to see fewer and fewer of those deleterious alleles, those harmful alleles, until finally they will actually all disappear. In other words, what happens is the mutation gets rid of its mistakes, or rather, to put it more correctly, natural selection gets rid of mutation's mistakes. And what you're left with is a gene pool that does not contain these harmful mutations. By the same token, these very, very few beneficial mutations also don't stay the same in frequency. They tend to increase in frequency. They become more and more common in a population because they help organisms pass their genes on until finally they're going to displace even the neutral alleles, and they're going to completely take over. Despite the fact that most mutations are harmful, organisms evolve to become better able to deal with their environment because harmful alleles go away and helpful alleles do the opposite. They increase in frequency. We'll learn more about that when we talk about natural selection. I want to talk briefly about some misconceptions that I often hear about when students write down things about mutation. Again, this is the peppered moth. Before the Industrial Revolution, this is what you would find if you went out walking in the woods around London. If you looked closely enough, you would see on some of these lichen-colored tree trunks, you might see a moth that looks like this. Or you might not see it because it's really well camouflaged. This is the little a phenotype, the recessive phenotype of the peppered moth. And you can see that that peppered color pattern blends in wonderfully with this kind of tree trunk. The melanic phenotype, not so much. This organism is very vulnerable to predators, particularly birds, which can see quite well. This was what was going on before the Industrial Revolution. The Industrial Revolution brought into being a tremendous number of factories that ran on coal. And as you can see in this picture, coal is not a clean burning fuel. Enormous amounts of soot were belched out of the smokestacks of London and other industrial centers in England during the Industrial Revolution, and it really changed the way that the forest looked. Lichens are very sensitive to air pollution, so most of them died, and the bark and practically every other surface in the forest got carpeted with this blanket of soot, so that after the Industrial Revolution, tree trunks started looking like this. Now the melanic form of the moth is very well camouflaged, and the peppered moth stands out as though it had flashing orange and red lights that said, "Come eat me." So this is the scenario. And this is the sort of thing that you often see students describe, or this is the way that you often see students describing it. So for example, I'll often see students write things like, the soot made the melanic alleles come along that were less visible. Or because dark-colored moths are hard to see, the moths mutated from gray to black. I'll also see statements like this. Moths needed to be dark-colored to survive, so they mutated from dark to gray. Or dark moths were harder to see, so the moths gave their offspring the melanic allele. All of these basically misdescribe what mutation actually does and why. The environment does not cause mutations, useful mutations, to happen. Whether or not there is soot on the tree trunks, the mutation from the little a alleles or the big A allele is happening at the exact same rate. Okay? Useful mutations don't happen because organisms want them, and they don't happen because they need them. Organisms don't affect the rate at which they mutate. They don't will new mutations into being. Mutations are just happen. They're accidents. Similarly, what's important to remember is that mutations are random. And what do we mean by that? Well, we mean that whether or not a new allele is going to be useful has no effect on how likely it is to occur. So again, when the forest looks like this, you get little a alleles mutating to big A alleles at, say, a rate of 1 in a million. Suddenly the forest changes, it looks like this, and you have alleles mutating from the little a to the big A form at a rate of 1 in a million. It doesn't change. Okay, the fact that this mutation is suddenly useful rather than harmful does not change the rate at which it occurs. What you want to do is be thinking about mutation selection in the following way. The first step is that mutations happen. They are accidents. They happen at random with respect to whether or not they're going to be useful. And then selection acts only after the mutation has occurred. Once there is a big A allele in a population of moths, selection can act to weed it out, in this case because it's harmful, or it can act to increase it in frequency because in this forest it's helpful. Okay, so this is a two-step process, and you don't get anywhere evolutionarily without both of the steps. But the mutations happen first by accident, they're random, and then selection acts. Selection is not random, selection is very systematic. And selection is what causes new mutations to change in frequency. That's what we'll be talking about next.